I want to talk about the last important group of organic molecules. That's the nucleic acids. And if you look here, there's three different models of a nucleic acid. You probably recognize the ladder structure as DNA, the center picture there. The one on the left is the actual structure where DNA is sort of a twisted ladder, like a spiral staircase. And then the space-filling model shows how the electron clouds fill in and give it that three-dimensional view. Anyway, nucleic acids, we commonly call them DNA and then RNA. So we'll talk a little bit more about DNA and RNA now. First off, these are the two guys that discovered the structure of DNA. Their names were James Watson and Francis Crick. They're kind of controversial. They stole some of their data from a woman and didn't give her credit. She died of cancer. But anyway, Watson and Crick are the two guys that you'll hear a lot about in terms of discovering the structure of DNA. The basic structure, the monomer of DNA, is this thing called a nucleotide. A nucleotide has three parts. It's got a phosphate group. We've talked about that important functional group, a phosphate group. And it's attached to a 5-carbon sugar called a pentose sugar. That is then attached to a nitrogen base. Nitrogen base could be one of several different bases. Some of them are single rings. You can see up here the pyrimidines are a single ring, such as cytosine, thymine, or uracil. And then some of them are double rings. They're called purines. Those would be adenine and guanine. Okay? The pentose sugar in DNA is deoxyribose. You can see it there. In RNA, it's ribose, and the only difference is, you notice on RNA that it's got a hydroxyl group there. If you deoxygenate the ribose, you get deoxyribose. So that one hydroxyl group is the only difference between the deoxyribose and the ribose sugar. So anyway, this nucleotide is the basic unit that makes up DNA and RNA. In terms of DNA base pairing, we always pair adenine with thymine and guanine with cytosine. Easy to remember that is A's and T's are both made with straight lines. G's and C's are both made with curved lines. Okay. Now, why does A only pair with T and G only pair with C? Well, there's two reasons. First off, structurally, we need every rung of the DNA to be about the same size. If you're building a ladder, you want every rung to be the same size. So we need to have a double ring with a single ring. Okay, we can't have two doubles or two singles, or our DNA won't fit together right. Okay. The second thing, notice the hydrogen bonds that cause these base pairs to bind together. Okay. We have to have the hydrogen and oxygen, or hydrogen and nitrogens, lined up just right so that we can form those hydrogen bonds. Those hydrogen bonds are really important because that allows this helix to stick together, but hydrogen bonds are also very weak, so they can very easily be unzipped, and we can get at that genetic instruction to make RNA and a protein, or to replicate the DNA and use that strand as a template. So these weak hydrogen bonds are really important, and you can see A's and T's line up these hydrogen bonds nicely, as do G's and C's. Okay, uracil is in RNA, and it pairs up with adenine. So in RNA, we'll see A pair with U. And we'll get a little bit to that in a little bit later. Again, the golden rule of DNA, why DNA is the blueprint of life, is that it contains the code for proteins. So you can think about DNA as these recipes. The recipes on DNA are basically genes, genetic instructions to make a protein. They get copied into RNA. That RNA then travels outside the nucleus to the ribosome, where it is translated into a protein. So DNA makes RNA, makes protein. That's the golden rule of genetics, and it's an important concept in biology. So this is just a quick overview of nucleic acids, our last organic group.